I'll introduce our next generation leader speaker, Neville Sanjana. Neville got his, attended Stanford for his bachelor's, and he got 2 degrees. One is Bachelor's of Art in English Literature, and the second is Bachelor of Science in Symbolic Systems. I've heard of this symbolic systems major at Stanford. It's a very unique major that's associated with some famous female technology giants. Correct me if I'm wrong, Marissa Mayer, Yahoo CEO, also got a bachelor's degree in symbolic systems from Stanford. So as you can see, Neville is already very technology-driven from the very beginning. After his undergrad study, he went to MIT to get his Ph.D. With Sebastian Song. And he did some nice work in the project that's preceding Sebastian Song's interest in connectome. So he worked on electrophysiology in cultured neurons and trying to understand the computational functions of those cells in the culture. After that work, I think he did a short stint in George Church's lab because he's also interested in a molecular aspect of the neuroscience research, or in general, biological research. And from there, he actually met Feng Zhang, and immediately became good friend with Feng, and actually very soon became Feng's first postdoc when Feng started his own lab at the McGovern Institute and the Broad Institute. And together with Feng, Neville has done some great work in developing the latest genome engineering technologies, including both TALEN, the TAO technology, and the CRISPR technology in just a short 3 years of period. And now I think Neville has some interest in applying those technologies into addressing some really interesting biological questions by constructing large-scale genome engineering libraries using the CRISPR technologies and trying to understand the interactions between genes, circuits, cell functions, and things like that. Welcome, Neville.
Thank you so much for that introduction. That was a really nice introduction, actually. And thanks to the Allen Institute and the organizing committee for inviting me here. I'm really thrilled to be here, actually. Since first seeing the brain maps that the Allen Institute produced years ago, I've been wanting to visit and be a part of this kind of exciting adventure. So I'm a postdoc in Feng Zhang's group right now at the Broad Institute and also at MIT. And I'm going to talk today about genome-scale CRISPR-Cas9 screening. And you'll see that I'm going to put a few different projects together. So I'm calling this phenotype to genotype and back again. So what's been clear in genomics over the last decade, and actually even longer, is that there's just been this amazing advance in genome sequencing technology. Our ability to read out genomes and exomes has just grown exponentially. And so I'm just plotting here, this is a figure from the National Human Genome Research Institute, at the number of GWAS papers and publications. And you can see just even, this is only up to 2011, but the increase is just absolutely humongous. And so as we've been able to sequence more and more patients, we've been able to find more and more human gene variants and also non-coding variants that are associated with disease. But the ability to write DNA, the ability to perturb the genome, has really lagged far behind the ability we have to read DNA. That's just been an amazing technological story, but the flip side of the coin, the writing of DNA, really hasn't kept pace. And this writing DNA is actually quite an important ability to have, because what we'd like to do is take these disease-associated variants and really say something about causality. Do these variants that we find in these patients, because the genome is huge, there's many variants in each patient, do they really cause the disease? We'd like to take healthy cells, healthy organisms, put in a variant, and really say that causally link this gene of interest to a particular phenotype. And so today I'm just going to focus on a few different approaches to doing this, and you'll have to kind of indulge me. There'll be some neuroscience near the end, but there'll also be some other stuff. So basically, there's really two ways we can look at this, right? We can take a hypothesis-driven approach, focus on a few genes of interest, and try and figure out all the phenotypes that are associated with those gene variants. This is a very common approach with making a model cell line, model organism. The other approach that we can take is to focus on a phenotype, a disease-related phenotype, and say what are the genetic elements that underlie this phenotype? Can I somehow do an unbiased screening approach to find these? And so I'll talk today about two different projects for causal gene discovery. The first one is a genome-wide screening approach in human stem cells and human cancer cell lines. The second one is an isogenic modeling approach with very specific mutations that have been implicated in de novo forms of autism spectrum disorders. And looking at phenotypes in, we just had a nice introduction from Jonathan, a friend of mine from MIT, looking at these in a different kind of human cortical tissue, maybe not quite as realistic as what Jonathan's using. So for the genome engineering toolkit, and just a little background to get us all on the same page, what we really mean when I'm saying writing genomes, being able to put in causal variants, is facilitated in many cases by targeted nucleases. And the reason we need targeted nucleases is that they enable us to, at specific locations in the genome, create double-stranded breaks. And there's a variety of double-strand break repair mechanisms, but in mammalian cells, the two relevant pathways to a lot of the genome engineering work that's happened in the last few years is these two pathways here, the non-homologous end-joining pathway and the homology-directed repair pathway. And so you can think of this one here as kind of the more error-prone pathway, and this one as the quote-unquote perfect repair or templated repair pathway. And so actually a lot of the work I'm going to talk about today is going to focus on this pathway, which is actually massively more efficient in mammalian cells. And so here what happens is basically if you target a double-strand break to, say, a coding region of the genome, there's a little bit of, a bunch of enzymes are accrued there, there's a little bit of chewback of the DNA, and then it's very important for the cell to fix, as you can imagine, any chromosomal break. And so if this double-strand break is targeted to a coding region of the genome, to an exon, let's say, there's a little bit of either chewback or a few bases are added, creating an indel mutation, triggering a frame shift, a premature stop codon, also nonsense-mediated decay. There's all these quality control mechanisms in the cell. Basically, this is a way of enabling gene knockout. On the other side here, we have homology-directed repair. This is templated repair. This is where, in addition to this double-strand break, there's some repair template, either exogenously added by an experimenter or off another chromosome. And here, what can happen is we can take advantage of this to be able to stick in a LOXP site, a GFP tag. Many of you are probably very familiar with homologous recombination. This is that repair pathway. So in our lab, I really feel like I've been very fortunate to join Feng's lab at a time where there's been a huge amount of technology development. And some of the earlier work with CRISPR was spearheaded by two grad students, Le and Anne, in the lab. And they really pioneered this very particular system, bringing it into mammalian cells, which is the Cas9 CRISPR system. And I'm going to explain a little bit more about the details of how the system works, but it's been quite a revolution in biology because it's just a very efficient system for doing multiplexed genome engineering. And really to understand why CRISPR has had the impact it has, you have to start with where I started my postdoc, or even ideally much further back, to see where we were kind of in the pre-CRISPR era. And so pre-CRISPR, we were kind of on this side of the slide here, where we were using these kinds of tools. And in fact, one of these, the tail nucleases, is what I worked on first in my postdoc, a very relatively new technology still. Both the zinc finger nucleases and tail nucleases, which are fusions of DNA binding domains and enzymes to create those double-strand breaks. The major thing to know about these, without getting into too many details, is that in these, a protein specifies the DNA targeting. So all these tools are programmable DNA targeting reagents. So these are all like pointers to the genome. But here, you have to do protein engineering. In this case, the unit of modularity is each zinc finger binds 3 bases of DNA. With talons, each tail repeat, each 34 amino acid repeat binds 1 base of DNA, so a little bit finer scale modularity. But in both these cases, you have to do some protein engineering to reconfigure where you're binding in the genome. So as we think of scaling up to all these human disease variants, doing tons of protein engineering, even though there are tricks around this that we and others have used, it's still just not a fun thing to do. So the quantum leap forward with CRISPR-Cas9 really is that it's not the protein, the enzyme that creates the double-strand break, Cas9, this yellow blob here, is generic. It doesn't change its structure. What is changed here is a small RNA piece, this sgRNA or single guide RNA. And I'm just going to go over, because this might not be such a genome engineering audience, so I'm going to go over kind of the specifics, but maybe many of you are already familiar. So the RNA, these bases in blue, these 20 bases, when this RNA is loaded into the Cas9 enzyme, it directs Cas9 to, in this giant genome, in a complicated genome like the human genome, it can direct Cas9 to a region of the genome that has base pairing, that matches this 20 base sequence of this RNA at the DNA level. And so what's required is these same 20 bases here, and also what's known in the CRISPR biology field as the PAM, the protospacer adjacent motif. In the case of all the work I'm showing you from Streptococcus pyogenes Cas9, this is a bacterial enzyme is where this comes from. For Strep pyogenes, the PAM motif is NGG. So basically, you're able to target anything that's programmed into these 20 bases of this RNA sequence followed by NGG, so fully a 23 base pair targeting sequence. And this guides Cas9 to locations in the genome to create those double-strand breaks that then enable the genome engineering. So the real, as I said, quantum leap forward here is that we have RNA targeting, not protein engineering to do. And so with another postdoc in the lab at the beginning of 2013, this postdoc, Ophir Shalom in Fung's lab, and I started thinking about using DNA microarrays to actually make large-scale libraries of these sgRNAs to target Cas9 to many locations in the genome to really take advantage of this RNA targeting versus protein targeting. So to go back to this diagram, this kind of work where you can synthesize large amounts of these sgRNAs, instead of making one model organism or 10 knockout mice or 10 knockout cell lines, what we really wanted to do is show the ability to focus on a specific biological phenotype or disease-associated phenotype and look at all oops, that skipped one more forward. Look at all genetic elements that underlie that phenotype. And this is actually not a new idea. There are many pooled screening approaches that already exist. Ooh, that's fast. Such as pooled shRNA libraries that have been around for more than a decade now. Pooled libraries so you can, shRNA is used to knock down genes. There are also pooled libraries of overexpression constructs to overexpress or upregulate genes. But one thing that's important to point out with this genome engineering approach versus these previous pooled screening approaches is this right here, which is that when you're working at the DNA level, you have access to a whole lot more than just knocking down or playing with transcripts. You can access the other 98% of the genome, the non-coding genome, and also elements that aren't even transcribed at all, that just are DNA elements, but still can have function. And so after tantalizing you with talking about screening the non-coding genome, I'll say that I'm going to focus the rest of the talk on just screening the coding genome, but you should keep this possibility in the back of your mind because I think this is where a lot of the action is going to be over the next few years, especially with psychiatric disease genetics. So here's an overview of kind of what we did to make this genome-scale CRISPR knockout screening library, which we gave the name GeCKO to. So we use these DNA oligonucleotide array synthesizers to synthesize a large library, thousands of sgRNAs, clone them in mass into a plasmid library, package these plasmids in lentiviral format, and then transduced different cell types of interest with this genome knockout library. And I'll explain more about the choice of lentivirus actually in a second. After transducing this in this pooled format, we can then enrich for whatever phenotype we're interested in, whatever disease-associated phenotype or just normal biological phenotype we want, and then by deep sequencing, read out which gene knockouts are enriched and which ones are depleted from this library pool. And so just before I get into the pooled screening, I'm just going to talk a little bit about why we use lentivirus. There's two major reasons why lentivirus is a nice delivery technique here. One is that we can control the multiplicity of infection, which is really important for us to be able to make sure that each cell in the pooled screening approach only gets one knockout reagent. We don't want to have to disentangle the effects of multiple gene knockouts in a single cell. The other reason is that lentivirus kind of is its own barcode for deep sequencing. Because lentivirus is an integrating virus, it makes it easy for us to be able to kind of read out the screen by just PCRing from this sgRNA cassette. And so I told you kind of earlier about the components of the CRISPR system. So here again is that we have kind of a ubiquitous promoter just expressing the Cas9 protein. Along with that, we have a puromycin resistance gene that's expressed with Cas9. That's so we can just, so again, it's a very low MOI infection. We want to be able to select for the cells that got the constructs themselves. And then there's also, driven by a Pol3 promoter, the sgRNA cassette. So again, Cas9, the CRISPR system, we're using the protein component, the enzyme Cas9, and a short guide RNA sgRNA component. And so just showing you how this knockdown kind of system works, the easiest comparison is, of course, with something that a lot of you have used in your labs is shRNA, which takes advantage of the RNA interference pathway. And so what I'm showing here in green is HEK cells, so human cells that have one copy of GFP integrated into the genome. And here in gray are wild-type HEK cells, so these are just regular HEK cells. And these are 6 different sgRNAs directed against GFP. And you can see that in most cases, the phenotype here, when you FACS sort these cells, is absolutely indistinguishable from wild-type cells. So these are sgRNAs that we've put into, so we've transduced the GFP cells with lentiCRISPR carrying sgRNAs targeted against GFP. Here we've done the same thing with some of the 4 best shRNAs available at the Broad RNAi platform against GFP. And this axis is kind of misleading. You know, this comparison looks like, oh, these guys didn't work. But if you look at this is a log scale. This is actually, all these sgRNAs have over 90% knockdown. These are excellent sgRNAs. But what I'm trying to do here is draw out the difference between knockdown of a gene and complete knockout. And so sure enough, if you deep sequence, so if you deep sequence at this locus, you can see that almost 100% of the GFP alleles are modified just after 7 days of expression of this lentiCRISPR construct. So knockdown versus knockout, and I'll come back to this later. So here's just a little bit about designing this library, because it's kind of nice to know the process of what goes into this. So what we wanted to do is we actually wanted to look over the entire genome and pick out ways of knocking out specifically each gene one at a time, but not affecting. We didn't want Cas9 to have off-target activity at other genes in the genome, so we could be as certain as possible about unique targeting. And so what we did is we picked early exons of these genes that were constitutively expressed. And so to measure the constitutiveness of different exons, because as neuroscientists well know, there's a lot of alternative splicing, a lot of different isoforms of many of these genes, we use the Illumina human body map data. So this is transcriptome RNA-seq data from about 20 or 30 tissues in the body. And so we just looked for exons in each gene that were expressed in as many tissues as possible. And so that gave us kind of this list of which are the constitutively expressed exons. And then in those exons, we went ahead and listed all Cas9 targetable sites. So this is basically any DNA sequence that has NGG, the PAM sequence that's required for Cas9. And from there, we wanted to prioritize this list of all possible, because there's a lot of, in each exon, as you might imagine, there's many sites that have the NGG motif. So which ones should we pick? Thankfully, at the time that Ophir and I were designing this library, 2 graduate students in the lab, Patrick and David, had just, not published yet, but had just gotten this beautiful data set showing on-target versus off-target specificity of Cas9 binding over many targets. And what they found is that the PAM proximal, the most 3' bases, mutations in these really disturbed Cas9 binding, versus at the 5' side, away from the PAM sequence, these mutations were much better tolerated. So the position of these mutations mattered. Another thing that mattered is the number of mutations. So if the site is unique in the human genome by 2 bases versus 3 bases, the 3 bases is a much better choice. So the more unique the site is, the less likely you would have Cas9 off-target activity. There's also a clustering, a center of density effect, a centroid effect, where if mutations are more spread out in this 20 base pair sequence, you're more likely to have cutting there. So several mutations closely spaced prevents this off-target cutting. So long story short, we took this empirically derived formula that they had found based on their examination of CRISPR activity in different sites in human cells, and basically took this into account for scoring our guide sequences and picked those with the least likely to have off-target effects elsewhere in the genome. And I'm happy to talk about more details about this too. So we then synthesized these, as I mentioned, on an oligo-array synthesizer. We cloned these into a lentiviral library. And so in our initial library, which has some shortcomings that I'll talk about later, we had about 65,000 sgRNAs covering 18,000 genes in the genome. So what we aimed for was a redundancy of about 3 to 4 sgRNAs per gene. And all these were cloned into that lentiCRISPR vector. Okay, so then what we did is we delivered these to cells, we applied selective pressures to those cells, and looked for enrichment and depletion of different sgRNAs. So one of the simplest screens you can do is simply a negative selection screen. So in this setup, what we did is we took the 2 different human cell lines, A375 cells, a cancer cell line, and Hu62, a pluripotent human stem cell line, transduced them with the GeCKO library at a very low MOI, selected with puromycin, and then just waited for 2 weeks to see how did the representation of the library change between that initial infection and 2 weeks later. And what we found is that there's a dropout of many of the constructs in the library over those 2 weeks. So the complexity of the library drops down less as some of the sgRNAs are disappearing. And so when we asked which sgRNAs are actually disappearing over this time, the results are actually very unsurprising. They're exactly what you'd expect. The ones that are most depleted are sgRNAs that have to do with absolutely essential processes in the cell. So things like the structural elements of the ribosome, RNA processing, RNA binding, RNA splicing, polymerase II. These are genes where if you knock out these genes, the cell absolutely cannot survive. And so this is a very nice validation, initial validation, that the pooled library screening approach is working as we might expect. And there's a good amount of agreement between these 2 cell lines, even though they're quite different biologically. So the next type of screen that we wanted to do was a positive selection screen. So instead of seeing what depletes, we can look at what enriches. And so for that, we turned to a nice cancer model cancer problem. And so in melanoma, there's most malignant melanomas. The most common mutation found in malignant melanoma is a missense mutation in the BRAF gene, this V600E mutation. And so this has kind of been one of the early discoveries in cancer genomics, in sequencing genomes of cancerous tissue. And so there was a very nice effort over about 8 years to develop a very, very targeted therapeutic to specifically attack mutant BRAF. And if you think about this, this is actually kind of amazing, right? There's 1 amino acid different between this mutated form of BRAF and the normal form of BRAF. And so this drug, vemurafenib, and some others developed at the same time, was approved in 2011, 2012 by the FDA, and it has incredible specificity for just the mutated form of BRAF. And so this was thought to be kind of like a silver bullet, kind of just like magic therapy. And so what you initially see in the clinic is actually quite impressive. So here's the melanoma patient with these tumor nodules you see all over the body. And then after 15 weeks of treatment with vemurafenib, the remission is very impressive. Unfortunately, in almost all cases of vemurafenib treatment, this was just discovered in the last few years by Levi Garraway's lab at the Broad, you see the cancer come back, and if you look to see the locations where the tumors are, you'll see that they've reoccurred in very similar locations to where they were initially. So what's likely happening is that there's a small group. Most cells are dying due to vemurafenib's exquisite targeting of mutant BRAF, but there's a small group of cells that are resistant, that are being harbored there, and then given a chance to grow back. And so we wanted to ask with the GeCKO library, can we find some of the mutations that lead to vemurafenib resistance? And so what we did is we took A375 cells, which have biallelic V600E BRAF mutation, and we transduced them with the lentiviral library. We selected just at a very low MOI. We selected for a few days with puromycin. And then for the next 14 days, we either treated with vemurafenib or just with a vehicle control, just DMSO. And then we harvested a portion of the cells that we were culturing at 1 week and 2 weeks to read out the representation of the GeCKO library and to see what we could find. And just kind of the gross phenotype, as you'd expect, the cancer cells that are untreated just double out of control. They grow hugely. And the ones that are treated with vemurafenib, it's pretty static, and then you can see the population drops a little bit here. This is still, as in the previous figure, would be kind of the initial treatment phase. And so if you go ahead and deep sequence that library, what you find is the representation is quite stably maintained over 2 weeks in the vehicle treatment, basically the untreated cells, because they all keep growing. But with the vemurafenib treatment, starting at day 7, but really it's quite clear here at day 14, you see a sudden drop in the sgRNAs that are preserved in the library, and that's good. That's vemurafenib killing most of the cells that are in the experiment. But what you do see is you see a few sgRNAs that are highly overrepresented. So this is just count of those different sgRNAs, the different CRISPR targeting reagents. And this group clusters quite distinctly from the rest of these conditions. And of course, the natural question is, well, what are these sgRNAs that are taking over the cell culture? So what I've plotted here is the frequency of the sgRNA in the day 14 vemurafenib, just counts of how many times we see that sgRNA in the day 14 vemurafenib condition versus how many times we see that same sgRNA in the day 14 vehicle condition. You can see that there's this cloud of dots that's kind of above the equality line. And if you start to label these dots by which gene is being targeted, you can see here, for instance, these 4 red dots are distinct sgRNAs. So these are completely different sequences, completely different target sequences in the genome, but they all target this gene, NF2. Same thing here, these distinct orange dots all target this gene, MED12. So that we were really happy to see this. So we can take this sgRNA-based view, and using algorithms that have been used previously for RNAi screens, we can collapse it down to a gene-based view. And when we do this, we see a few genes that seem to have significance much higher than other genes, the other genes in the library. And 2 of these genes had just been recently validated by other groups. So MED12 had been shown to confer vemurafenib resistance by René Bernard's group in this very nice Cell paper. And earlier, so this screen that we did was based on we essentially copied an shRNA screen that had been done at the Broad just a few months earlier, published in this paper, a really nice paper by Stephen Whitaker. And the main hit they found from their screen was a gene called NF1. So we recover NF1 and MED12 from these 2 existing vemurafenib papers, and then of course a few other hits, which I'll show you some validation data for in a second. But we were very lucky, I think, to have this RNAi screen, this genome-wide RNAi screen that had been done on the same cells with the same treatment, because for CRISPR screens, was kind of our first of its kind, and so we wanted to be able to make a comparison with a tool that's well known, well understood by a large community. And so I'm just going to draw out this comparison a little bit more, because I think it's very nice to see the difference between these screening methodologies. So here on the top, I'm plotting the CRISPR screen. On the bottom, I'm plotting the genome-wide RNA screen that Stephen and Levi Garraway's lab did. And what I'm showing here is the top 10 gene hits using this gene enrichment, the collapsing to the gene level score. And both papers use the exact same algorithms developed at the Broad. And of these top 10 hits, what are the fraction of reagents that are enriched as the top 5% of enriched sgRNAs in the CRISPR screen, or the top 5% of enriched shRNAs in the RNAi screen? That's kind of confusing terminology, but hopefully this makes sense. So what you see is a huge difference in consistency. So here, their top 10 hits, not all of the hairpins, not all of the shRNAs are enriched. In fact, very few of them, like overall like 25% or something. And this is a genome-wide screen, and I'm just showing you the top 10 hits. These are the ones we expect to have the underlying biological reagents to be greatly enriched. But here in the CRISPR screen, we see something quite different. For almost all of these top 10 gene hits, in fact, we didn't actually validate all of these genes, because we only got about to number 7 in the list, and then we said, okay, this is enough. But from almost all of them, you see almost all the reagents for each gene, almost all the distinct sgRNAs are in this top 5% of enriched sgRNAs. And this large difference in consistency between the CRISPR pooled screening approach and the RNAi pooled screening approach really suggests to us that large scale, so the Allen Institute does a lot of large scale projects. There's similarly at the Broad Institute, we have large scale cancer genome screening projects using pooled shRNA screens, such as Project Achilles, to find the Achilles heel of each cancer. So we found that this difference, even though it's only in one screen, might suggest that this screening approach really has a large competitive advantage over the kind of the dominant screening paradigm right now for loss of function knockdown screens. So just to kind of draw out the comparison with one of the genes that was found in the CRISPR screen but not identified in the pooled shRNA screen, this gene NF2. So if you look at applying these at CRISPR reagents targeted toward NF2 versus shRNA reagents targeted toward NF2, look at the protein level. It's actually pretty similar. Both of them look quite good. Like there's this band seems to go away. But when you look at the phenotype, things are drastically different. With the different sgRNAs, you see log orders increase in vemurafenib resistance. With the different shRNAs targeting NF2, only one of them seems to show some change in vemurafenib resistance. And I think this gets back to what you saw on the GFP targeting slide, where really this is the difference between knockdown and knockout. With complete knockout, those cells that have complete knockout are able to really proliferate and take over the population. But in this case, just partial knockdown might not be enough to confer this resistance phenotype. And of course, if you, similarly to what we saw with GFP, if you deep sequence after either 7 or 14 days, you see almost all the NF2 alleles are modified. There's almost 100% indel rate. And so what we did is we went ahead and took several of the other hits from our screen and validated them individually with sgRNAs that were both in our library and novel sgRNAs targeted toward these genes, and we were able to validate them with CRISPR, and then some of them validated also with shRNA. I swear I'm a neuroscientist, I'm not a cancer biologist, but it's worth just saying that these are not unexpected. These are not random hits in the genome. They fit into the biology of BRAF actually quite nicely, several of these. So NF1 directly regulates an upstream RAS enzyme. CUL3 is an E3 ligase that's known to ubiquitinate NF1. MED12 is involved in the TGF-beta pathway. NF2, one of the new hits from our screen that we really focused on, is involved in several growth pathways, such as the mTOR pathway and the Hippo pathway. Some of the other novel hits from the screen, TATA1, TATA2B, these are all transcriptional adapter complexes that work along with polymerase. in this STAGA complex. And these are some of the main drivers of oncogenes like MYC. So these hits make sense within the context of cancer biology. I just like showing this slide because it's good to see some of the raw data too. And I just want to, for those that are not familiar, just want to show you that this is not, when I say gene knockout, this is not just like one mutation that we're seeing on the genome. There's a huge range of indel sizes. So this is just deep sequencing data. And what I'm showing you is different sizes. Negative numbers mean deletions. Positive numbers mean insertions at the location of the cut. And so we see many different mutations made through these reagents. So this is individual application of some of these different sgRNAs from the library. I often get asked with genome engineering about off-target effects. People are very worried about off-target effects. And in some of the work I'll show a little bit later in the talk, this is something important to worry about. But for pooled screening, we really don't care, actually. Off-target effects are of really no care. And that's because we're not relying on a single sgRNA, really, to target the gene. We have many sgRNAs voting for each gene. And it's really the consistency of their voting that enables us to say this gene is a true hit for vemurafenib resistance. This gene is not. There's just one enriched sgRNA. But still, it's worth seeing that even with this lentiviral overexpression of Cas9 and the sgRNA constantly in the cell, that we actually get very high levels of on-target modification, essentially 100%. These are the most likely off-target sites computationally predicted in the genome. And here for NF1, we see almost no modification. For NF2, there are a few of the sgRNAs do have some off-target modification. But again, for the pooled screening approach, this is not really that worrisome. In fact, if you are worried, there's a very simple solution. Just put more sgRNAs in your library. Speaking of which, we do have, on the next slide I'll show that. But so one of the problems with the screens we did is making the virus was just a huge pain. So this virus, because it carries this large Cas9 transgene, turned out to be very low titer, and initially we made no effort really to optimize it. So we had to just make vats of this stuff and ultracentrifuge it to actually get it into volumes that we could really handle. And so this was, when we presented this, it wasn't easy to tell other people, well, now you should go do your own pooled screens, and hopefully you have an ultracentrifuge to enable that. So one thing that we did is we went back to the bench and really re-engineered the lentiCRISPR vector. We just took every piece of it apart and tried to do what we could to improve the titer. And so with this lentiCRISPR version 2, you'll notice the vector map is basically the same. A lot of the changes are under the hood here. And what we were able to do is boost the titer tenfold from the original lentiCRISPR that we used for all the screens I just showed. In addition, we realized that for a lot of the screening applications, like the one I showed with the cancer cell lines, you really don't need to deliver this large Cas9 with the library. So we broke the system into now a two-component system, also increasing the biosafety of this genome-modifying system so that you first can create a cell line just with Cas9 in it, make this a stably expressing cell line, and then later deliver the library as a separate guide-only library. And this one has about 100-fold increase now, this lentiGuide Puro over this. This enables you to do we now routinely do pooled screens just with supernatant virus. So this is just a huge speed up to our screening process. Okay, thanks. So there's a lot of issues with the first library that we made, the human library, that we wanted to improve on. So we recently made the version 2 GeCKO library for the human. A lot of people working in mouse models asked us, well, can I use this library in mouse? No, you can't. So we also made a mouse library. So these new libraries have absolutely uniform coverage of even more genes, each one with 6 sgRNAs targeted. We've added this is the only bit of non-coding biology I'll talk about. We've now added microRNA targeting. So we have 4 sgRNAs per microRNA. We've tried to cover almost all the microRNAs in mirror-base for both the human and mouse genomes. Big omission from the initial library is there were no control sgRNAs. We now have 1,000 sgRNAs that don't target in either the human or mouse genome. And both of these are available now in the lentiCRISPR-2 and the lentiGuide Puro that I just showed. So just to summarize this part of the talk, I showed that lentiCRISPR cuts at a DNA level and that there's really is this difference between knockout versus knockdown. And that this GeCKO pooled screening approach shows a lot, at least in the one melanoma screen that I presented, shows higher consistency in the screening reagents than sgRNA. And because of this, we were able to find both all the already known vemurafenib or PLX resistance mechanisms and also some novel ones. And that we just recently created these new libraries and new vectors that should hopefully make doing pooled screens much easier in the future. And if there's anything I guess you take away from this talk is that working on the genome, there's a much larger space of possible genetic manipulations. And I think this work just kind of scratches the surface, but I think there's much more good stuff hopefully to come. So with that, just want to thank Ophir is a postdoc in Feng's group that did all the genome screening work with me and really was just a partner 100% in this work. With the electrophysiology and some of the recent stem cell work, I've been working with Cici and Tongyi Liu, also at the Broad and MIT. And Ella was a very talented technician in the RNAi screening platform that helped us with some of the genome-wide screens. And of course, my advisor, Feng. The lab is bigger than this now, but this is an old picture. Feng has just been an inspiration kind of in everything I do, and I'll take some questions. Thanks.